our question and answer session. As a reminder to the audience, if you would like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. Please hold for a brief moment while we pull for questions. Paul, you may begin when you're ready.
I think actually we already answered the question that was, will we be presenting your results at AACR? Yes, we will. I'll take that. And then, so this question came in, it's please comment on the types of solid cancers that express TEM8. So I think Mihnea presented a little bit on that, but Mihnea, if you could take that.
Can you repeat the question? Sorry, I was...
No problem. So the question was, is that please comment on the types of solid cancers that express TEM8.
Yeah, there are basically 2/3 of the database where we look at mRNA expression for TEM8. It's more than 1,000 types of cell lines, and about more than 60% of them, they express TEM8. And all the cells which were checked, actually, they express TEM8, and they are permissive to SVV.
Alrighty. So the next question is, please comment on the long survival in mice with SVV-001 and checkpoint inhibitors. What's the likely mechanism? So as Aman was talking about, he gave some of this, but basically the mechanism is that when you combine SVV with checkpoint inhibitors, you get a systemic anti-tumor immune response. So that's essentially why the tumor on the other side of the mouse, that's why that actually tumor was eradicated as well as the initial tumor. So that's what's going on, and that's that's one of the wonderful things about essentially what's called the immuno-oncology revolution, is that these things are becoming understood. And so we're doing a lot more studies. We have a lot of mechanistic data on this aspect, and we'll also be presenting this at the ACR. So we have to talk about that further if you'd like. Then the last question is just comment on the ARM constructs with SVV-001. Well, I talked a bit about that. We're very excited about this program. So as you already saw from Aman, that presented some of this work, I mean, the GFP expression is very selectively expressed in the tumor, and we've done this in the relevant murine model. So we're definitely very excited about moving this forward. We've got a large platform of therapeutic genes that we're able to actually produce, manufacture, and we're very excited. We're going to be very excited about sharing some of that with you coming up pretty soon. Any other questions? Let's see, those are active questions. Okay, so if I go to all questions, I think I can see that there's some more questions coming. Let's see. So I think Aman, did you cover what the clinical endpoint is? That's a question that came in. Aman, would you mind taking that?
Yes, thanks, Paul. That's an important question. Neuroendocrine cancers are a little unique in the way we design these because of the indolent course of the disease. However, median PFS is the industry standard to assess the effectiveness in the phase 2 part. So the primary endpoint for us is to look for the median PFS of Seneca Valley virus in combination with checkpoint versus just the checkpoint alone for the control arm. For the phase 1, the primary endpoint is to look for the toxicities, the SAEs, DLTs, the dose-limiting toxicities, and come up with the safe recommended phase 2 dose.
Thank you. So another question is, can you elaborate on the cancer types that Seneca Therapeutics might address with SVV, in addition to the neuroendocrine tumor and neuroendocrine carcinomas that Aman talked about? So we actually are very excited about, have some interesting data in triple negative breast cancer, as well as sarcoma. So there's actually, quite a wide variety, but when you look at, these are really super unmet medical needs. Those two indications, they can also be treated with intratumoral administration, which is very good. And if you look at our 8 gene set on those indications using slides, essentially FFPE slides, the vast majority of those cancers should be permissive for SVV. So those are 2 out of many. And then we'd also, as we're doing a randomized trial, we're going to do commercializing manufacturing lots to bridge into that study. And at that point, we think we'll be ready to go back to intravenous administration. And we're starting to think about the types of indications that we'd want to go back into for that. But we may want to even try intravenous administration in terms of bridging into that the study that's ongoing. And as Aman was talking about, 60% of cancer is pretty wide open. But we have a lot of data on a lot of indications, even using that 8 gene set now. So, let's see. I think right now, I'm just looking to see if there's any other questions. Okay, I think we've got. So I think this is all the questions that have come in. So I just want to thank you very much for everything. I guess we can stay on for a little bit, but right now there's no more questions. So I really want to thank everybody for attending. And feel free to reach out to us if you have any additional questions. And hope you enjoy the presentation and thought it was interesting. Anything else?